I'd like to first say thank you to everybody in the audience for taking time to listen to the Luminary story. I'll give you a distillation of our company, and typically what I have done in the past is being part of a company that's founded by three scientific genome engineer renowned people. Our slides are usually packed with a lot of technical data, and I try and parrot them and pass myself off as a scientist. So I'm not going to do that today. I'm going to tell you more about the business side, which is an area that I'm a little more comfortable in. So effectively, what our company is focused on is obviously patient care. We are very focused on antigen escape and T-cell dysfunction. And we think by doing that, we will get less relapse and more recovery. So that's kind of our one vision of our company. We're pursuing three markets with two assets and two manufacturing platforms. The markets that we're pursuing is a market that's currently a commercially existing market, non-Hodgkin lymphoma, and there's some big players in that, but we think we can disrupt that market, and I'll explain more about that later. We then think we can take the same asset and move that over into the autoimmune, which I will also explain. And then we have a universal receptor platform that we think will be well set to work in the solid tumor space. So from a manufacturing platform, we come at this different from a genetic modification. We use a non-viral system that we developed at a company we had called Beamogen, which is called TC Buster. And then we also have an allogeneic platform that we think is unique and different than anything else in the market. Finally, the assets that were being developed is a BAF CAR that is a ligand-based CAR that effectively has 3 receptors on it, which is what we think will help deliver results that effectively mitigate antigen escape. And then I'll talk about our North Star platform. So in essence, this is our company. So the markets that we're pursuing, as I mentioned, are existing commercial markets, at least in the first market. And we believe that 1 in 4 patients will fail traditional CAR T therapies due to antigen escape, where the CD19 receptor on the cancer cells will stop expressing itself on the cell surface, and therefore those patients will relapse. We'll talk about our technology and why we think that will not happen with our technology. But if we're able to get a 5% market share of today, what is a $1 billion market, that would mean we'd have $50 million in revenue. As I think everybody in this audience probably knows, those treatments are moving from 3rd line to 2nd line, which will triple the patient number. So we have a $150 million market potential, we believe, if we get 5% market share. We're going to take that same asset, this BAF CAR, and we believe that it can play in the autoimmune disorder, and specifically we're going after systemic scleroderma. And then with our universal receptor, we think we can play in the solid tumor, which we all know is a very underserved market today with CAR T therapies. So we do have an IND approved already. We've only been around for 2 years. And one of the reasons we were able to do that is because of our manufacturing expertise, which I'll also explain. We're going to have 4 different dosing strategies. Obviously, the primary endpoint is safety, and then we hope to see some telltale signs of efficacy. We plan on treating 12 to 18 patients. The clinical trial will be done at Cleveland Clinic. So the team, this is what I want to kind of emphasize most, because I've worked with this team now for 5.5 years. We had a company called Beamogen Biotechnologies, which was a tools company, and we successfully created a transposon system of the likes of Sleeping Beauty and Piggyback, but made it available to the industry. Today, we get 60 to 70% integration rates with this technology. These guys that are beside me here are just some of the best genetic engineers I've ever seen in my life, so I'm just proud to work with them. And we have what we call, you have to have a GSD degree to work at Luminary, which is get shit done. So we're really big on our cell manufacturing. Between my experience at working at a CDMO and Bo Weber's experience at bringing a couple of CRISPR technologies into the clinic, we think this is one of our expertise. I'll do a very quick plug here for my friends at ScaleReady. We very much believe in the G-Rex product. But what we do is we use this TC Buster system to genetically modify our T cells. And the three big things about that are the fact that we can get larger cargo in, we can get it done at a very cost-effective rate, and most importantly, we can get GMP reagents in 4 months, as opposed to the wait you have at Lenti when you're a small biotech startup. Again, we have an allogeneic platform, and what makes us different is we use gamma delta cells, which get rid of the HLA match issues. But what's different about our platform is the fact that when we have our engineered platform, the engineered gamma delta cells, we can turn around and expand those cells up to about a 2,000-fold expansion rate. And additionally, we carry both populations of the V1 delta and the V2 delta. So everybody else in the gamma delta space typically pick one or the other of these gamma delta cells to use. We have the benefit of bringing them both to our final product. This is our pipeline. As I said, we have an IND that's approved that we're getting ready to start a clinical trial, provided that we can get funding to do so. We will follow up very quickly with an allogeneic platform to do the same type of work, but we'll be able to do that in a very quick fashion because the manufacturing process is the same. The CMC section is already written. All we have to do is an additional tox study. We will then turn around and take that same BAF CAR and we'll work on using that for systemic scleroderma, and I think I have some slides in here to explain why we think that's applicable. And then we have this universal receptor program that I'll talk about in a bit. So one of the benefits of our BAF CAR is the fact that it can touch on many different areas. So obviously we're going to start a basket study in non-Hodgkin lymphoma and work on mantle cell cancer patients more specifically. I'll talk about the autoimmune. I did just get an email yesterday that I was pretty excited about in regards to myeloma. I got an email from a gentleman at Case Western who said that there is a whole host of manufacturing issues with the current people that have CAR T therapies for myeloma. And at the Moffitt Cancer Center, there's over 200 patients that are on a wait list. So he wanted to know if we would do a trial because our BAF CAR has, with the antigen receptors on our CAR, which I'll explain in a bit, have applicability to myelomas. So again, back to the GSD degree that you have to have at Luminary, here's what we've done in a very short time frame. You can see that we in-licensed this BAF CAR in June of 2020, and in March of 22, we already have an IND approved. And we already have our clinical grade GMP reagents, or we'll have the last of them delivered to us in another month. We've done this on a shoestring budget, which is what we're kind of known for. We're known for being very capital efficient at all the companies we've had in the biotechnology space. So now to the BAF CAR specifically, and here's where I'll try and pass myself off a little bit as a scientist. So the current challenge with CD19 therapies is up to about 25 to 30% patients are relapsing between 4 to 9 months once they're treated with a CAR T therapy. The whole reason is that the CD19 on the cancer cell stops expressing on the surface. So we have a CAR that's different. It's not a single chain base CAR, it's a ligand based CAR, and it has 3 different receptors on it: BCMA, BAF, and TACI. So we have a strong belief and a feeling that, in fact, the cancer cells are not going to be able to, stop expressing all 3 of those particular antigens on the cell surface. And we have all kinds of data that shows we only need 1 of them to get cancer killing. So our universal receptor program is, rather than engineering a CAR or a TCR onto T cells, we engineer on this particular receptor, which allows us to effectively put arming molecules once the T cells are engineered through an infusion, and we can change those out at will. We can turn them on, we can turn them off, and then effectively we can change that particular antigen. So this gives us benefits in regards to antigen escape. It also gives us benefits in T cell exhaustion, because there's literature that shows today that if in fact you rest those T cells for a while and then re-stimulate them and reactivate them, they in fact have more rigor to control and start killing the cancer. So one of the things that's different about, and there's 3 or 4 companies that have tried this before, and so far nobody has really made this successful, but one of the things that is different about our kind of universal receptor, universal CAR program is the fact that we use the TCR signaling as opposed to the CAR signaling. So we've co-opted that and have that working in a platform. So I think one of the things that I missed to talk about was systemic scleroderma. So I've got a little bit of time left here. I will kind of go back and cover that off. But for systemic scleroderma, if you're not familiar with that disease, it's a B-cell mediated autoimmune disorder. And those pathogenic B cells that are in those patients express BAFF and TACI. So therefore, we have a strong belief that our particular technology will take care of that. And a benefit of our BAFF CAR is we do not wipe out all of the B cells. So some of the early naive B cells are preserved with our particular therapy as opposed to CD19 that wipes them all out. So we are currently undergoing experiments where we have the full plethora of B cells that we're doing our BAFF CAR testing in vitro on. And then we expect to work with some key opinion leaders to be able to take patient samples of blood and PBMCs and effectively treat those blood samples in vitro again with our BAFF CAR. We know there's not a good animal model in mice for systemic scleroderma, but it's a disease that once it gets into your lungs and you have fibrosis of your lungs, it is lethal, and today there are no treatments for that. So again, we're out trying to raise money for our company to be able to do this clinical trial for non-Hodgkin lymphoma. And if anybody has any ideas or interest in potentially helping us get this clinical trial done where we think we will stop 1 in 4 patients from not having a relapse due to antigen escape, I would love to talk to you. Thank you for your time.